Hello, good evening, guys. Good evening. How are you all doing? Am I audible to everyone? Can anyone write it on the chat box if I'm audible or not? Good evening, Aman. Good evening, Selvapriya. Good evening. Okay, I'm audible, right? Good evening, everyone. Good evening, Ahmed. Good evening. So today I've come up with the CSIR NET 2021 paper, which was held on 17th February. So we'll be discussing about the questions that came from Unit 3, from both Part A and from both Part B and Part C. Okay. So So the first question, which came, like we'll be doing for shift 1 and shift 2, both of the papers. And starting with shift 1, the question ID was 29th, which asked basically, in bacteria, many of the tRNA genes do not contain the CCA sequence found at the 3' end of the tRNA. In this context, which one of the following statements represents the correct explanation? Now, this was kind of medium question, like everybody would have, if they would have studied, no, that the translation chapter in Watson itself, it is being given regarding this in a table, in a tabular form. It is being given. The explanation is being given regarding this. So, how many people attempted this particular question, and how many did it right? Can I know it? The options here are, in these tRNAs, amino acylation occurs at the 3' end of the tRNA, irrespective of the presence of the CCA sequence. Second, it says that the CCA sequence is added to these tRNA transcripts in a DNA template independent manner. Or the part C, it says these tRNAs exploit the process of tRNA splicing to include a CCA sequence at their 3' end. The absence of the CCA sequence occurred only in the last common ancestor LCA during the course of evolution, and the current day tRNA genes always possess a sequence to encode the CCA end of the tRNA. So which of the following options do you find correct? Did anybody attempt this particular question? Okay, Amit Kumar says he didn't appear. So whosoever appeared and attempted this question, can you please write on the chat box which particular option that you opted. Nobody attempted. Okay, no problem. The correct option for this particular question is option B, right? So we'll know how. The explanation has been given here. The 5' CCA to 3', this particular end is universally conserved for all tRNAs and is absolutely required for protein synthesis, right? This everybody knows. The 3' terminal trinucleotide CCA 3' to which an amino acid will be attached during protein synthesis is present from few bacterial and all eukaryotic tRNA precursors, right? It is present in few bacteria and in all eukaryotic tRNA precursors, and it is added during the processing. So the genes that encode tRNAs, they do not encode the CCA end, right? When this particular tRNA is being synthesized, at that particular point of time, this CCA end, 3' CCA end, it is not being incorporated, right? There is a specialized RNA polymerase which is required to incorporate this CCA end. This is called as CCA adding enzyme, right? It is a tRNA nucleotidal transferase. It adds the terminal CCA to the tRNAs that initially lacks the sequence, and there is no nucleic acid component to this enzyme. So it basically acts as a template independent manner, right? So there is no requirement of, or there is no nucleic acid component to this enzyme, that is like CCA adding enzyme add a specific sequence to the end of the tRNA without an RNA or DNA template. That is why this particular option, option B, is correct, that CCA sequence is being added to these tRNA transcripts in a DNA template independent manner, right? So this particular option would be correct for this. So did you understood like how this particular option would be correct? Yes. Janet, okay. Option D, you said option D is not correct for this particular question. Option B is the correct one. So explanation was being given here, and it was being explained how that CCA sequence is being added to these tRNA transcripts. So when the tRNA transcripts, it is being synthesized. At that particular point of time, this CCA sequence present at the 3' end, it is not actually present in the 3' end, right? It is being incorporated later on by a specialized polymerase, RNA polymerase enzyme, right? Which adds CCA at the 3' end. And it adds the CCA at the 3' end in a DNA template independent manner. It doesn't require any DNA template or RNA template to add this particular sequence, right? So option B would be correct for this particular question. Now coming to question ID 31, it is for shift 1 I'm talking about. So the question here is, it says, all of the following statements about the bacterial transcription termination are true except. So you will need to give an answer which of the following statement is not correct, right? Incorrect statement you have to find out. Now, who all like they attempted this particular question? Those who those whosoever have given the exam, whosoever have attempted this particular question, can you write down in the chat box which particular answer would be correct for this question? Yes, I would be inviting the answers. Okay, Amit Kumar, it's not a problem. You can see this particular lecture later on also. It will be there in the YouTube. So can anyone answer this particular question? What would be the right option for this? So options are: some terminator sequences require Rho protein for termination, inverted repeat and T-rich non-template strand define intrinsic terminators. Rho-dependent terminators may possess inverted repeat elements. NUS A is necessary for intrinsic transcription termination. Which of the following options would be incorrect? Okay. Nazia Shafi, she said answer D. Yes, Nazia, you're absolutely right. Option D is the incorrect one. Rest, all of the other options are correct here. So that for explanation, here is the explanation for this particular question. So it says that the intrinsic termination, also called the row independent termination. So there are basically two types of terminations: intrinsic termination and row dependent termination. So intrinsic one is the row independent termination, right? So here it says that it involves the terminator sequences within the RNA itself. So the mRNA which is being synthesized, within that mRNA itself, there will be a terminator sequence, right? Which basically acts as a signal. So the RNA polymerase, it basically acts as a signal for RNA polymerase to stop during the transcription termination, right? This terminator sequence is usually a palindromic sequence. It's a palindromic sequence that forms a stem loop hairpin structure, right? Because of that, only the termination takes place. So following the stem loop structure is a chain of uracil residue. So this stem loop structure is there. After that, there is a uracil residue. There is a multiple uracil residues would be present in there. Since the bonds between the uracil and the adenine are very weak, so the adenine would be present in the DNA, while this uracil would be present in the pre-mRNA which is being synthesized, right? So the bond between the adenine and uracil, it would be very weak bond, which will lead to the dissociation of the RNA polymerase from the DNA template, right? So this is how the termination takes place in case of intrinsic termination, right? Now, the uracil stretch in RNA corresponds to the T-rich stretch in non-template DNA strand. So this much is clear, right? How intrinsic termination basically takes place, right? Then comes about the extrinsic termination, which basically utilizes the termination auxiliary factor. So here a row factor is being required, right? So in extrinsic termination also, all the sequences, the signal sequences which are present in intrinsic termination, that would be present in the extrinsic termination, but there would be an extra factor would be required, that would be the row factor, which will basically enhance the termination, right? So this row terminator may include other factors also like tau and NUS-A, right? So NUS-A is basically present in case of row dependent termination, not row independent termination. So based on these above facts, what we can say that the statement 1, 2 and 3 are correct. However, the statement 4 suggests that the NUS-A is necessary for the intrinsic transcription termination, which makes it as an incorrect statement, right? So option D would be incorrect here. NUS-A, it is basically being required in row dependent termination, not in intrinsic termination, right? So option D would be the incorrect one here, right? Did everybody understood this particular question? Type yes. Now coming to question ID 32. So this particular question says that which one of the following proteins is essential for both the initiation of the DNA replication as well as the continued advance of the replication fork. So here it is basically the proteins that it shows here. It is saying that the proteins, they are essential for the initiation of the DNA replication as well as it is being required for the continuation or advanced replication fork, for making of the advanced replication fork also. So all those proteins which are required, you have to like correct that particular option here, right? So first one is ORC, right? Second is geminin. Geminin is basically a DNA replication inhibitor which binds to the CDT1 loader. So in eukaryotic replication, there is like loader protein is being present. One of the loader protein is the CDT1. So this loader CDT1 protein, it is being inhibited by the geminin protein, right? So geminin is a kind of DNA replication inhibitor here. This is something which is very new for all of you. So you can add up this particular pointer in your notebooks because this is something which is new, which you found here, right? So ORC is something which is required at the initiation, starting of the replication, right? So then comes the CDC45 and the other option is CDC6. So which one of the following options is correct one? Which one of the following options says that this protein is responsible for the advancement as well as the initiation of the DNA replication? So can anybody answer which one would be the correct option here? if you have attempted this particular question. Okay, Bhava Priya, she says option C is the correct one. Okay, anybody else? Anybody else have attempted this particular question? Yes, so Miss Priya, you are like you have attempted the right. Yes, it's the right option. Option C is the correct one. CDC 45, right? So how it is? So we'll be doing it. We'll be knowing it through the explanation here. So CDC 45 and GINS protein, both of these proteins, they bind to the MCM 27 helicase. So MCM 27 helicase is the helicase protein which is required for the unwinding of the DNA in case of eukaryotic replication, right? So CDC 45 and GINS proteins, these two proteins are the one which binds to this helicase enzyme in case of eukaryotic replication and during eukaryotic replication initiation, and they together form the CDC 45, MCM 27 and GINS complex, which we basically call as CMG. It is majorly known as the CMG complex, right? This is the active form of the MCM 27 DNA polymerase, right? DNA helicase, right? So this complex is basically the active form of helicase, which is required for the unwinding of the DNA. Unless and until this pre-initiation complex is not being formed and the unwinding of the replication fork doesn't take place, the further replication process won't be taking place, right? And this helicase complex is responsible for the unwinding of the DNA during the initiation and continue the advancement of the replication fork. And this complex is also required for the continuation of the replication fork, right? The continuation of the replication going on into the DNA. So these two proteins are the one which you should have marked. So CDC 45 is the correct option here. So option C is the correct one. So whosoever marked option C are right. Okay. So Salva Priya, you are also right. And Nazia Safi, you have also marked the right answer. So good. Right. Then coming to the option, the question ID 33. So it says, which one of the following combinations represents the major protein or protein complex involved in chromatin condensation in yeast and human respectively, right? So anybody have answered this particular question can mention their answers in the chat box. So the question is asking which one of the following combinations represents the major protein or protein complex involved in chromatin condensation in yeast and humans. It is talking about yeast and humans here, right? So anybody attempted this particular question? Yes? No one? Okay, no problem. So we'll be reaching to the explanation. So what does this particular question ask? So firstly, let me tell you the option B is correct. So whosoever attempted this question, so option B would be the correct here, SIR complex and HP1 complex respectively in case of yeast and humans. So these complex would be responsible for the chromatin condensation, right? So how the two proteins are responsible? So HP1 is basically known as the heterochromatin protein. It is a conserved component of the highly compact chromatin of higher eukaryotic centromeres and telomeres, right? So chromatin protein 1 is a non-histone chromosomal protein. It is a non-histone. So there are two types of proteins involved. Right? There is a histone protein involved and there is a non-histone protein involved. So it comes under the non-histonic proteins, right? They are primarily associated with the heterochromatin regions in Drosophila. So in case of Drosophila, these HP1 are basically present and involved in the formation of heterochromatin regions in case of Drosophila, right? The gene encoding HP1 and SUVAR25 is a dosage-dependent regulator of the heterochromatin, silencing in Drosophila. So wherever these heterochromatins are present, they basically silence the gene, right? there the replication, transcription, translation doesn't take place. So these particular these proteins are responsible for like for assistance of this particular heterochromatin region, so that the gene silencing takes place. So in budding yeast, heterochromatin establishment is achieved by targeting of the CSIR protein complex to telomerase or silencers at the silent mating type locus through the DNA binding proteins, where SIR2 deacetylase, the H4K16. So this is very important here. So firstly, it says that the heterochromatin protein 1, it binds to the H3K9 trimethylase. It participates in the chromatin condensation, right? And SIR is basically a silent information. It gives the silent information. It is a regulator present in yeast. That what does it do? It basically deacylates the H4K16, which will participate as the heterochromatin. So all these things are this SIR is basically responsible for the condensation, right? So here the option which is correct is basically option number B, the SIR complex and HP1. Both of these are responsible for the chromatin condensation. So formation of heterochromatin regions, they are responsible for basically for the gene silencing in case of yeast in humans. So in case of yeast, SIR complex is responsible, and in case of humans, HP1 complex is responsible, right? So option B would be the correct one here. Okay, everybody understood the explanation, right? Now moving on to question number, the question ID 51. So this particular question says that the basic difference between the direct repair and the base excision repair is. So like all of you would have studied about these two repair systems, right? In case of like in molecular biology. So there are two types of direct repair and base excision repair. The basic difference it has been given here. So you have to choose the option which says the correct one. So first option says the direct repair restores the original structure of the altered nucleotide without replacement, while in base excision repair, the section of the DNA containing the distortion is removed, the correct base is being added and resealed. So this particular option seems absolutely right. Okay, but further moving on to the other options also we'll be saying. So option B, it says that the direct repair, homologous recombination repairs the broken region, while the base excision repair restores the original structure of the altered nucleotide by modification, right? So as we know that the direct repair, it is not connected to the homologous recombination repair. So the indirect repair is the basic example of the direct repair is basically the thymine diamond where it is being formed, right? Those kind of mutations which takes place. When it's basically the UV photo lesions takes place at that particular point of time, these direct repairs is being like involved in repairing the particular damaged DNA. So it is not linked to the homologous recombination. So this particular option would be incorrect. Coming to option C, it says that the direct repair restores original structure by non-homologous and end joining without using the homologous template, while in base excision repair, the section of the DNA containing the distortion is being repaired by using the homologous. So both of these, this option is also not correct, because as we know, the direct repair is not linked to non-homologous or homologous repair system, and base excision repair is also not involved here there. Indirect repair, option D, it says that the indirect repair, an exonuclease, a DNA polymerase, and a ligase are used, while in base excision repair, a translation polymerase that bypasses the bulky lesions is used by the cell. So this option is also correct. The option A is the correct one here, right? So Aman Jha, he gave the correct answer. Selvapriya also. So yeah. So this particular option A is correct here. So both of you are right. So explanation here says that a few specific lesions can also be removed by direct chemical reversal and inter-strand crosslinks. So inter-strand crosslink is another kind of mutation which takes place where basically intercalating agents are present. Mutation here takes place. So in this also, in this repair also, here also the direct repair participates, right? So, like small subsets of DNA lesions, like in case of UV photo lesions and alkylated bases, are simply reversed in an error-free manner. So there is no like error involved in this particular case. So error-free manner, these repair system takes place. So for BER, that is the base excision repair transactions, chromatin remodeling and the DNA damage site is followed by the lesion recognition by a DNA glycosylase. So here a DNA glycosylase enzyme is basically responsible, which basically so excision, the particular base which is like unwanted base, it is being excised out from the DNA sequence by this particular enzyme, DNA glycosylase, which can recognize and excise a damaged base from the undistorted helicase, as well as once flipped out from the major group. So from the major group, it gets flipped out, right? So option A is the correct one, which says that the direct repair restores the original structure. So original structure is being maintained here in case of direct repair of altered nucleotide without replacement. While in case of base excision repair, the section of the DNA containing the distortion, the section of the DNA consisting of the distorted base, it is being removed, and the correct base is being added, and it is being resealed again, right? So option A is the correct one here. So coming to the option ID 65. So this particular question says that which one of the following regions of the target gene is not used for making an RNAi construct to knock down its expression. So this particular question is basically related to the RNAi construct, right? So here gene silencing, how it takes place, all that stuff you need to remember. So whosoever gave or attempted this particular question, can they write down which option they have opted for in this particular question? So this is a very easy question. Even if you don't know about the RNAi, from the option itself, you can see like which particular option won't be required here. Okay, Selvapriya says option D. Yes. Anybody else? Anybody else who attempted this particular question? This is a very easy question. Okay. Bhava Priya, she also says it's 3. It's option D. Yes. So both of you are correct. Option D is the correct one here. Intronic regions. So intronic regions, whether they are present, like if they are present or not present, their presence doesn't affect the expression of the gene, right? So which one of the following regions of the target gene is not used for making an RNAi construct? So in this particular question, the option D is the correct one. So explanation for this is that RNAi needs to knock down the expression of a gene cannot be constructed using its intron, as they are removed during the RNA processing. So these intron regions, they are basically being removed during the RNA processing itself. So they are not being used for building the RNAi construct, right? So introns are these are basically the large chunks of the RNA within a messenger RNA molecule that interfere with the code of the exon. So exonic regions are the basically one which are the expressive ones, right? And these introns get removed from the RNA molecule to leave a string of exons attached to each other. So this everybody knows. So option A says the 5' UTR of the mature transcript. So 5' UTR and 3' UTR, these are the regulatory regions. So these are required very much in case of construction of the RNAi construct, right? So, and exonic regions, these are the coding regions. So these would be required. So intronic regions are the left one, which you need to strike this particular option, because this particular option is the correct one, where these intronic regions are obviously they are already being removed when the RNA processing is being taking place. So these are not being required to building up the RNAi construct. So option D is the correct one here. So whosoever of you marked the option D in this particular question have done it right. Right? So Aman Shah, he also says option D. So all of you are correct. Now, here is a quick reminder for all of you. So Shaurya batch has already been started from 4th of March. So here we are providing you with the facilities such as the RAID classes would be there by the CSIR NET experts. Interactive online classes would be there, 24/7 doubt solving via chat, and printed hard copies would be provided to you. Study materials would be provided, and there would be complete video backups of all the classes. Right. So if you want to know more about this Shaurya batch and how it is going and what all things are there, you can contact us into the toll-free number that is flashing on your screen, and you can also like mail us through the email, and our admission experts, they are the one who will be guiding you how to proceed further, right? So limit seats are there, so you can enroll right now. Now coming to the next question, question ID 84, which says that the mitochondrial protein synthesis is of prokaryotic origin, right? So mitochondrial protein synthesis is of prokaryotic origin, that we know. So it says that the following statements are being made about the ribosomes from bacteria and mitochondria. So these options are basically based on facts. If your facts are clear, you will be able to solve this in this particular question. So first option says that the bacterial ribosome consists of small and large subunits of 30S and 50S respectively, whereas in mitochondria of mammals, these subunits are of 28S and 39S, right? So this particular option is absolutely right. This is something which you need to remember. So this is a fact-based question. So if you if you read like standard books, even in Watson, Lodish, in all of these books, this particular statement is being given regarding the bacterial ribosome and the mitochondrial ribosomes, right, in mammals. Coming to the option B, it says that in the bacterial ribosomes, the RNA is to protein ratio is about 2:1, whereas in mitochondrial ribosomes, this ratio is usually 1:2. So this particular statement is being given in Watson. You can check it out. And this is also a fact-based option, and this particular option is also correct. Then it says that option C, it says that both the bacterial and the mitochondrial ribosomes consist of 30S and 50S subunits. This is not right, because mitochondria, they consist of subunits 28S and 39S, right? Then option D, it says that both the bacterial and the mitochondrial ribosomes consist of RNA and protein in the ratio 1:1. So whosoever attempted this particular question, can you tell me which option you marked? So option 1 is A and B only, option 2 is B and C only, option 3 is C and D only, and option 4 is A and D only. Which of the following options you marked? Can anybody give me the answer? I have only told you the answers itself, so it would be easy. Okay, Celopriya says option A. Option A or option 1? Yes, Aman. Option 1 is the correct one. A and B statements. These are the fact-based question, and option A and B are the correct one here. So explanation here, it is being given that in bacteria, the ribosome consists of small and large ribosomal subunits, 30S and 50S respectively, that everybody knows, right? Making the intact 70S ribosome. Mammalian mitochondrial ribosomes have sedimentation coefficients of about 55S and consist of a 28S small subunit and a 39S small subunit, right? Making it like 55S subunit in total. Then it says that another difference between the mitochondrial ribosomes and their cytoplasmic analogs is in the RNA protein ratio. So RNA protein ratio differs both in case of mitochondria and in case of bacteria. So in case of bacteria, the ratio of the protein, the RNA to protein ratio is basically 2:1, while in case of bacteria, it is for mitochondria it is 2:1. For bacteria, it is for mitochondria it's 1:2, while for bacteria it is 2:1, right? So this particular option would be correct. Option A and B are the correct one here. Then coming to the option ID 85. So in option ID 85, it says that the given below are a few statements about the lambda infection cycle. So this cycle everybody should be knowing, right? So first it says that the competition between the C1 and C2 gene products determines the establishment of lysogeny versus lysis. So when the bacteria has to decide whether it has to go through the lysogenic cycle or lytic cycle, so there are some key components in the gene which plays a role here, right? So accordingly, these options are being given. The first option is being given here. Second option says that the C1 binds to OR1. It is an operator region, right? OR1 first while the crow binds to the OR3 first. The option C, it says the crow binding to the OR repressor, C1 transcription. And the last option says the rich medium favors the lytic cycle because the C2 is protected from the cellular proteases. So which one of the following options is the correct one here regarding the lambda infection cycle? If anybody would have studied this lambda infection cycle, this particular question would have been easy for them. Okay. So Selvapriya says option B and C. Anybody else have attempted this particular question? Fabia, okay, option 1. Right. So both of you are right. Most of you people would have attempted option 1 now. If you would have studied about the lambda infection cycle, these two options are the correct one. So explanation here is being given. So here it says in bacteriophage lambda, it is basically a virus that infects E. coli, right? This everybody knows. Now, upon infection, the phage can propagate in either two ways. Either lytic cycle will take place or lysogenic cycle will take place, right? A lysogen is extremely stable under control, under normal circumstances, but the phage dormant within it is the prophage, right? So prophage is basically being formed when the viral DNA is being incorporated within the bacterial chromosome. It can efficiently switch to lytic growth if the cell is being exposed to agents that damage the DNA, right? If the DNA gets damaged in the bacteria, then this particular this lytic cycle, so lysogenic cycle, it will immediately get converted into the lytic cycle, right? The switch from lysogenic to lytic growth is called the lysogenic induction. The switch between these two states is mediated by proteins encoded by the bacteriophage genome. So there are certain proteins that are present in the bacteriophage genome which are involved in this switch which takes place from lysogenic to lytic cycle, right? It basically happens when the damage of the DNA takes place through UV radiation or other kind of mutagens, right? So there are basically two genes which regulate regulating proteins, which is synthesized by the virus. First one is the C1 repressor, which is also known as the lambda repressor, and other is the CRO, that is the control for repressor, and the other things. So these are the two repressor proteins which are basically responsible for the switching of the lysogenic to lytic cycle, which represses each. So these two proteins basically represses each other's synthesis, right? So here in the diagram itself, you can see. So let me choose a pen. Yeah. So here in the diagram itself, you can see that there is the C1 protein which is there, and there is the CRO gene which is there. So this will produce the CRO protein, and this will produce the C1 repressor protein, right? Now here it says the two alternative arrangements of the gene expression render the lytic growth or the lysogenic growth. So lytic growth proceeds when the PL and PR, these are the promoters in the bacteriophage genome. So PL is here, this is basically present in the left side, and this is present in the right side. So right hand side promoter and the left hand side promoter. These remain switched on while the PRM is kept off. So when the PRM is kept off, this PRM is basically the region which is present in between the PL and PR. So when the PRM is kept off and the PAL and PR, these two promoters are switched on, then the particular growth cycle which takes place in bacteriophage, it is basically the lytic growth. And lysogenic growth, it basically takes place when the PL and PR are being switched off and PRM is being switched on. The regulatory region of the promoter right, that is PR, and promoter left, each contain an operator. So both of them contains an operator, the operator right and operator left. So both these operator right and operator left respectively, they are present in case of promoter R and promoter L, each consisting of 3 binding sites for C1 repressor. So both of these operators, they consist of 3 binding sites for this particular lambda repressor, right? So in the lysogen, 2 C1 dimers are bound cooperatively. So in a cooperative manner, in the lysogenic phase, the 2 C1 dimers, they are bound to the OR1 and OR2, and OL1 and OL2 to prevent the transcription from PR and PL. So if the transcription is being prevented from PR and PL, then basically lysogenic phase will be there. And if this PR and PL gets activated, then only this lytic phase will take place, right? So when two C1 proteins, they are cooperatively bound to the operator R1 and operator R2, and operator L1 and operator L2, it basically prevents the transcription of promoter, left region promoter and right region promoter. The C1 dimer bound to the OR2 activates the transcription from the maintenance promoter PRM by a direct protein-protein interaction with the RNA polymerase. So when the C1 dimer, it is being bound to OR2, right? It activates transcription from maintenance promoter. So from maintenance promoter, it activates the transcription. As a result, what happens? This C1 has a low binding affinity for OR3 relative to the OR3 and the other operators. So CRO binds to the same three sites of the OR and OL as C1 binds, but does so within the opposite affinities of the C1. So when the CRO binds to the OR3, when this particular CRO protein, this is the CRO protein which binds to the OR3 region, it prevents the transcription of C1. So if the transcription of C1 is being prevented from PRM, right, this leads to the inducing of the lytic cycle. So this C1 repressor gene is basically responsible for the switching or for the lytic cycle to continue. If this C1 is being inhibited by the CRO protein, so if this CRO protein inhibits the C1, this results into the inactivation of the lytic cycle, right? It prevents the transcription of the C1 from the PRM, thus inducing the lytic cycle. So C2 is required for the activating C1 gene's strong expression and thus required for the lysogeny, not for the lytic cycle. So in this particular option, the option that is being given here, option B and option C are the correct one, which says that the CI binds to the OR1 first, while the CRO binds to the OR3 first. Right, and the CRO binding to the OR repressor represses the C1 transcription. If the CRO binds to the OR region, right, the right hand side operator, it represses the C1 transcription. As a result, if this C1 transcription doesn't take place, then what happens? The lytic cycle won't be taking place. So this is the whole explanation for this particular question. Now coming to the option, this particular question, ID 86. This says that the reproduction of the Phi2 174, right? A single-stranded DNA phage involves several steps. So here is the replication of reproduction, the replication of the Phi X174 bacteriophage is being given here. The statements are being given regarding the replication in this particular bacteriophage. So a few statements are being given below which explains the mechanism. So first statement says that the single-stranded Phi X174 DNA is converted into a double-stranded replicative form, RF. Second, it says the replication of the double-stranded replicative form results in the production of single-stranded phages, about 50% of which are sense strands, like positive sense phages, and the remaining are the negative sense phages. Replication of the double-stranded replicative form results in the production of only negative sense phages, and the option D, it says that the replication of the double-stranded replicative form results in the production of only positive sense phages. So this particular question, if you know how the replication of Phi X174 bacteriophage takes place, you would be like, it would be easy for you to attempt this particular question, right? So if you would be knowing that in case of this bacteriophage, basically the positive sense, this is a positive sense phage, right? So most of the replicative DNA strands which are being formed, they are in the positive sense form. So if anybody have attempted this particular question, can you tell me which of the particular option would be the correct one here? Choose the option that correctly describes the processes. So out of 1, 2, 3 and 4 option, which one would be the correct one? Did anybody attempt this particular question? Option A and D. Okay, Selvapriya says option A and D. Right? Anybody else? Option 4, Aman. Right? Yes. Yes. So both of you are right. Option 4 is the correct one here. A and D are the correct options here. So first option, it says the single-stranded this this particular bacteriophage DNA is converted into a double-stranded replicative form, right? So firstly, single-stranded is being converted into a double-stranded replicative form, and last option, it says that the replication of the double-stranded replicative form, it results in the production of only positive sense phages. So only positive sense phages are being produced, not the negative sense strands. It is being basically being produced during the process of replication, right? At the end, it is a whole. If combined, it is basically a positive sense phage. So the explanation here, it says that the during reproduction or the replication of this phi 174 bacteriophage, supercoiled single-stranded DNA is being replicated to produce the double-stranded replication form. Right. So single-stranded DNA is being replicated to produce the double-stranded form of DNA, which in turn produces the single-stranded DNA that is in positive form. So this in turn produces the single-stranded DNA of the bacteriophage, which is in positive form, which in turn generates more RF forms. More replication forms are being generated, and it generates nearly 500 or more viral particles inside the bacteria, and they are being released by the cell lysis. That is, the lytic cycle here takes place, and through cell lysis, these viruses are being released, right? So option 4 is the correct one here. Now, coming to this particular question, this question ID is 87 in shift 1, which says that which one of the following schematics depicts the potential relationship among the subunits 2O and 2A and 2B of RNA polymerase 2. So, identify the correct schematics. So, which of the particular schematic diagram it is showing that is the correct one? So, anybody attempted this particular question? Yes. Did anybody attempt this particular question? Option C. Okay. Bhavya says option C. So Bhavya says option A. Keetana says option C. Salvapriya says option B. Okay. Different answers are coming for this particular question. So let us see which one would be the correct option here. So option A is the correct one here. So whosoever marked the option A, they have done it right. So explanation here, it says that the mammalian cells contain two forms of RNA polymerase 2, right, which are being designated as 2O and 2A, that differ in the extent of phosphorylation within the C-terminal domain of their larger subunit, right. So phosphorylation of this larger subunit, which results in the conversion of the RNA polymerase 2A to 2O, kinases basically help in this phosphorylation. So 2A can be converted into 2O by the kinases enzymes, right? So here it is being given. Kinases enzyme is basically being responsible to convert the 2A form to 2O form. So this particular option is like wherever this kinase. So here also we can see that the kinase is being given, but out of these two, which one would be the correct one? So this we can eliminate here. These two can be eliminated because phosphatase and this protease are not involved. Phosphatase is basically like it removes the phosphate group, and it is not responsible for the conversion of 2A to 2B, 2O. So from option A and option C, we have to choose which one would be the correct option. So another statement, it says that the reverse conversion can be done by phosphatases. So reverse conversion, that is from 2O to 2A. This conversion can be done by the phosphatases enzyme that remove the phosphate group. A third form of enzyme, RNA polymerase 2B, is found in vitro and lacks the repetitive C-terminal domain. So here it says this, so 2B can be made from 2A or 2O by action of protease that will remove the C-terminal domain, right? So here it says the protease would basically be responsible, that there is another domain that is the 2B form, it is saying. So from 2A or 2O, if the 2B form is being formation is being conducted at that particular point of time, this protease enzyme is responsible. So phosphatase is not responsible here. Phosphatase is basically used to remove the phosphate group, right? So protease, it basically what does it do? So here it says that the 2A from 2A to 2 or 2B, if we have to make the 2B form, then what happens, the protease enzyme is basically involved here, which basically removes the C-terminal domain, right? C-terminal domain itself is being removed. As a result, the 2A and 2O, it will be converted into 2B. So protease enzyme is responsible here. So option 1, that is the option A, would be the correct option here, right? This option is the correct one. Then coming to the option, this question ID 89. So it says that the following statements are made with reference to the DNA replication, right? So first statement, it says that the camptothecin causes intra-strand and inter-strand crosslinks in DNA, leading to the stalling of the replication fork. So this is an inhibitory protein, which is basically responsible for the inhibition of topoisomerase 1, right? So topoisomerase 1, for the inhibition of this particular protein, this is the inhibitory protein. So if topoisomerase 1 is involved, so it will basically act on the intrastrand, not the interstrands. Like two strands won't be there will be only one strand which would be responsible. And second option, it says that so this particular option would be wrong, obviously, right? Then the second option, it says the prevention of the reinitiation of the DNA replication during the same cell cycle is mediated by the regulating the loading of the initiator complex ORC. So it says the prevention of the reinitiation of the DNA replication during the same cell cycle, right, is mediated by the regulating of the loading. So this particular option is also incorrect, right? This is not the correct one. ORC is not responsible for the to prevent the reinitiation of the DNA replication. Now, see option C, it says that the A-glu to alanine mutation. So glutamine to alanine mutation, it takes place in the nucleotide building pocket of the DNA polymerase 3, which leads to the incorporation of the ribonucleotides in the extending DNA chain, right? So we will be coming to the explanation, we will be knowing how option C and option D are the correct one here, right? So like whosoever attempted this particular question, let me tell you option C and D are the correct one, and whosoever have opted for option C and D have done it right. Okay. Okay, fine. Don't be biased. I'll be speaking slow. So the explanation here, it is being given. Firstly, so as I have mentioned you that this particular inhibitory protein, that is the camptothecin, is basically, it causes the S phase cytotoxicity by producing the DNA double strand breaks, right? Concentrated in the replicating DNA. So statement A is the incorrect one here. Coming to the second statement, which this particular statement says, the prevention of the reinitiation of the DNA replication during the same cell cycle is mediated by the regulating the loading of the initiator complex ORC. So this for this statement, it is being written here that the prevention of the reinitiation of the DNA replication during this same cell cycle is mediated by the regulating the loading of the core replicative helicase. So this core replicative helicase is basically responsible, not the ORC, for regulating this prevention of the reinitiation DNA replication, right? So statement B is incorrect here. Now coming to statement C, how statement C is correct. So it says the DNA polymerase have a narrow active site suitable for the DNTPs. So DNA polymerase 3 is capable of discriminating between DNTPs and RNTPs. It is basically is able, due to its ability. No, it there are certain amino acids, like discriminative amino acids, due to which they are able to differentiate between DNTPs and RNTPs, and they only bind to DNTPs, not RNTPs. So it says that DNA polymerase have a narrow active site suitable for the DNTPs that would not easily accommodate the larger subunit at the 2' position present in the RNTPs. So in 2' position, which is like in RNTPs, that is the OH versus H. So here it basically says that the RNTPs at the 2 prime position, there are presence of the large substituents are present here. As a result, it is not able to accommodate these RNTPs into their active site. So most DNA polymerase use a bulky amino acid residue like glutamine called the steric gate, which is located at the entrance of the active site of the active site to prevent the RNMP incorporation. As a result, RNP incorporation doesn't take place because of these amino acids, these discriminatory amino acids, right? So reducing the size of the side chain. So as a result, the side chain, the size of the side chain, it gets reduced, of the steric gate reduced, increases the tolerance of the ribonucleotides in the active site. In some cases, to the extent of being incorporated as well as deoxyribonucleotides. So basically, because of the presence of these certain discriminatory amino acids, glutamine, right? Glutamine having the side chain, and it is basically a bulky amino acid. So if it is present in the active site, there will be no space for the RNTPs to bind in the active site of RNA polymerase. So there is a very narrow space in case of RNA polymerase active site, where it can only incorporate DNTPs, not RNTPs, right? So because of this reason, this particular option C is correct. So in option C, it says that if the glutamine, this amino acid, it gets converted or muted to alanine. Alanine is like it consists of the side chain, which is having it is not much bulkier as compared to glutamine, right? So it is a small size amino acids. So if it is present there, as a result, what happens? The DNA polymerase 3 it can incorporate RNTPs. So this particular option C is the correct one here. And coming to option D, for option D, the explanation is being given here that the topoisomerase T is an essential enzyme that is required for virtually every process that requires movement of the DNA within the nucleus or the opening of the double helix, right? This everybody knows. So this enzyme helps to regulate the DNA under an overbinding. So under an overbinding of the DNA helices, for this enzyme is basically responsible for, and it removes the knots and tangles. So entanglement and the tanglement, for the removal of these two particular things, this topoisomerase 2 is basically responsible in both the strands, not in one strand. Like topoisomerase 1 is responsible for like this entanglement and this removal of knots in only one strand, while topoisomerase 2, it leads to the cuts in both of the double-stranded DNA, right? From genetic material. So a mutation in the gene coding for topoisomerase 2 will basically lead to the entanglement of the daughter strands during the DNA replication. So because of these two statements, these two explanations, it is being said that the statement C and this option D are the correct one here. So option C and D are the correct one here, and option A and B are the wrong. So coming to, so this much has everybody understood how this particular option would be correct. So here option 3 would be the correct one. So whosoever are marked option 3 are right. Then coming to question ID 115. So here it says that a group of transposable elements describes as, they are being described as retro elements. It basically encompasses. So here for this particular question, there is a whole table that is being given in Watson, right? Page number, I guess, 409. So table number 12.2. So in that particular table, it is being given. So here the option A, it says that the P elements in the Drosophila lines but not shines in case of humans. B, it says that these are the retro elements. It is being shown that P elements, the copia and the P elements. So in which particular organism these retro elements are present? There's a whole table that is being given in the standard book Watson. So you can check it from there. So another option says the copia element in Drosophila shines but not lines in case of humans. Copia elements in case of Drosophila lines as well as shines in case of humans, and P elements in case of Drosophila lines and as well as shines in case of humans. So as we know that lines and shines, both of these elements, these retro elements, these are present in case of humans, right? So in option C and D, it is being given that lines and shines are present in case of humans. So out of these particular options, we have to choose which one would be the correct one. So coming to the explanation here it says there is a table, and this table I have picked up from the Watson itself. So Here it is being given two types of retrotransposons, the virus-like retrotransposons and the poly-A retrotransposons, right? So here it says that in the copia, it basically present in case of Drosophila, and line and shines elements are present in case of mammals, right? So from the option itself, what we can say, which option would be the correct one here? The copia element in case of Drosophila and the lines and as well as shines in case of humans. So this particular option is correct. So option C is the correct one here, right? Yes, Bhavapriya, you have done it right. Then coming to the question ID 169. So in question ID 169, here you have to arrange the DNA fragments in order of the decreasing melting temperature. So this question is very easy one. This though I guess everybody would have done, right? So can you please mention which one of the particular option would be correct here? Which order is the correct one? Yes, I'm waiting for the answer. If you have attempted this particular question. Okay. Amala Deepa, she says option number A. Right, means option number 1 you're saying. Right. Anybody else who have attempted this particular question? This is a very easy question. Yes, so explanation for this particular question is being given here. The nitrogenous bases are the purines and pyrimidines, everybody knows it. The purines are the adenine and guanine, while the pyrimidines are the thymine and the cytosine. The purines pair with pyrimidines by the hydrogen bonds, adenine pairs with thymine with two hydrogen bonds, while in case of cytosine, it binds with basically pairs with guanosine with three hydrogen bonds, right? So the melting temperature Tm, which is it talks about the basic definition of the melting point has been given here. It defines as the temperature at which 50% of that same DNA molecule species form a stable double helix and other 50% have been separated into single-stranded molecules. Right? So this is the particular Tm temperature at which particular point 50% of the DNA is present in the double helical form and 50% of the DNA is present in single-stranded form. Now, in comparing the DNA fragments, what we can see, the lengths are equal. Like we can see that the length of these DNA fragments, they are equal, but where is the difference? The difference basically comes in the composition of the G and C sequence. So G and C sequence, as we know that they are they are the complementary base pairs which binds to each other through three hydrogen bonds, and like melting this three hydrogen bond it takes a lot of energy, right? So in DNA fragments, A to D, A has six GC pairs. If you would be like comparing between A, B, C, and D, you would be finding that there is a difference of like 6 GC pairs in case of A, there's like 14 GC pairs in case of B, and there are 10 GC pairs in case of the C sequence, and in D there are 12 GC pairs, right? So B is having the maximum number of GC composition. So as a result, first is the B1, then comes D, and then comes C, and then comes A. So A is having the least amount of GC composition. As a result, melting this particular DNA fragment would be easier as compared to the B1. So option 1 is the correct one. The order is B, then D, then C, then A, right? Now coming to the question ID 170, right? So this particular question, it says that which one of the following statements about the short interspersed nuclear elements shines is true. So first statement, it says that the shines, it represent a class of retrotransposons. Second statement, it says that the shines can transpose immediately, independently shines, and shines can mobilize the neighboring line repeats. Shine are normally transcribed by RNA polymerase 1. So which one of the particular option is the true regarding shine sequences? So anybody who have attempted this particular question can answer me in the chat box which option they marked for this particular question. Okay. Don't be biased, he says option A, right? Bhavya, okay, option A. So this is the easy one, right? So option A is the correct one here. Shines represent a class of retrotransposons. So the answer for this is option A. So the explanation here, it says that the shines are non-autonomous retrotransposons. So only option 1 is the true one here, right? These basically represents a class of retrotransposons, and these are basically present in case of humans. So both shines and lines, as we have studied earlier, also the table that I have showed in one of the questions, there it was being written that these shines and lines, both of these retrotransposons, they are present in case of humans, right? Then coming to the option, this question ID 175. So this is this is from the shift 1 itself. So it says that a mutation in which one of the following sigma factors may be a possible cause of E. coli failing to adapt in response to thermal stress. So which one of the following sigma factors is basically responsible, and it is basically being induced in case of thermal stress? In case of thermal stress, E. coli, it can adapt to that kind of stress due to the presence of one of these sigma factors. So which one of the sigma factors is basically responsible for it? So if you have attempted this particular question, can you write on the chat box which option would be the correct one here? This is basically regarding this. Okay. Keetana says option B. Option B. Okay. Bhava Priya says option B. Don't be biased. Option B. Okay. All of you are correct. Option B is the correct one here. So explanation for this particular question is being given here. So it says that the sigma 32 is a specificity factor that is required for the normal expression of the heat shock genes and for the heat shock response in case of bacteria, right? So therefore, the mutation in it will make E. coli unable to adapt in response to the thermal stress. Coming to sigma 70. Sigma 70 is one of the first sigma factor which is being found in case of E. coli RNA polymerase. It basically acts only in initiation, right? It is basically responsible for the initiation, initiation to take place. But the transcription initiation to take place, but also it at an early stage of elongation to induce a transcription pause, right? And simultaneously to allow the fast lambda gene Q transcription anti-terminator to act. So here also it plays a basic role, this sigma 70, right? But it is not involved in heat shock or that stress condition to regulate the E. coli to regulate E. coli in case of heat shock, right? Then coming to the E. coli appears to have approximately 30 sigma factors, right? 30 sigma 54. This particular form of sigma factor, it has been found that in E. coli it has 30 form of sigma 54 dependent operons, right? And about half are involved in nitrogen assimilation and metabolism. So this particular sigma factor, sigma 54, it is basically responsible for the nitrogen assimilation and metabolism. And around 30 of these sigma factors are present in case of E. coli, it has been found. So for the particular question, option B is the correct one. These are the extra information regarding the sigma factors that I have given to you, right? So you can note it down somewhere. So now coming to the question ID 177, right? So in this question, it is a match the column based question where you have to match column A with column B. So here the given set of enzymes are being given in column A and the enzyme activities are being given in column B. So you have to match with them. So which of the following options would be the correct one here? So if you would have attempted this particular question, can you tell me which option would be the correct one here? So as we know that DNA polymerase is basically involved in single-stranded nicking, and DNA polymerase 2 it is basically involved in double-strand this break and ligation, right? So for A and B, it is being clear that B it is option 3. So wherever for B it's given option 3. Like in these particular options, it's given B for the 3 option is being given, and for A it is being given option 4. So for A option 4 also, yeah. So this we can eliminate, and from these we can choose. So here it says C2. So this polymerase epsilon, it is basically involved in the leading strand synthesis, right? It is involved in the leading strand synthesis. This here it should be delta, I guess. Yeah, by mistake I've written it, so you can correct it. It is delta. Polymerase delta is there. So polymerase delta is basically responsible for the synthesis of Okazaki fragments, the lagging strand. And while polymerase E, it is basically epsilon, it is basically responsible for the synthesis of the leading strand. So option A is the correct one here, right? So you can correct this particular this D option. By mistake I have written epsilon. It should be epsilon. This polymerase delta. So polymerase delta is basically responsible for the synthesis of the Okazaki fragments, that is the lagging strand. So option A is the correct one here. The explanation says that the topoisomerase are of two general types. The topoisomerase 2, it basically makes the transient double strand breaks in case of DNA, through which they pass a segment of uncut duplex DNA before resealing the break. And type 2 topoisomerase require the energy of ATP hydrolysis for their action. So type 2 requires ATP hydrolysis, and it basically cuts in both of the strands, right? It makes a cut in both of the strands of the DNA. While type 1 topoisomerase, in contrast, make transient single-stranded breaks in DNA, allowing the uncut strand to pass through the break before resealing the nick. In contrast to type 2 topoisomerase, type 1 topoisomerase, it doesn't require any kind of energy in the form of ATP, right? So this is the basic difference between topoisomerase 1 and topoisomerase 2. And DNA pol delta. So here it is being given here. So the last option, it was DNA polymerase delta and epsilon are specialized to synthesize different strands at the replication fork. So with DNA polymerase epsilon, it synthesizes the leading strand, while the DNA polymerase delta, it basically synthesizes the lagging strand. So option A would be the correct one here. So whosoever have marked option A have done it right. So coming to option ID 225. So here are the following statements were being made about transposomes. So again, one of the questions it again came regarding the transposomes. So the statements here says that first statement it says the transposomes have inverted terminal repeats, and their integration generates inverted repeats at the flanks of the target site in the host genome. Then it says that a composite transposome can transpose as a unit. C option, it says that the transposition event may cause deletions or inversions or move a host sequence to a new location. And last option, it says that the replicative transposition proceeds through co-integration. So out of these options, which options are the right one? So anybody who have attempted this particular question can tell me which option they have marked. Okay, Baba says she hasn't attempted this particular question. No problem. I'll be giving the explanation for this particular question, which would be the correct option. Keetana says option 2. Okay, Keetana. Anybody else? Amala Deepa. Option 1. Right. So we'll be proceeding and we'll be coming to the explanation of this particular question. So let us see which of the option is the correct one here. So option 2, whosoever are marked option 2 have done it right. So option 2, it basically says B, C and D. So these three options are the correct one here. So explanation, it says that most transposable elements, it generate flanking direct repeats on each side of the point of insertion. So wherever the point of insertion is there, in this point of insertion, in both the sides, there is the flanking direct repeats are there in case of most of the transposable elements, right? And many transposable elements also possess terminal inverted repeats. So at the terminal, there are inverted repeats which are present, right? And they cause the genetic changes. So option B, it says that a composite transposon can transpose as a unit. So it can transpose means it can jump or it can move from one place, one locus to the other locus. As a unit, it can move, right? Then it says that the transposition event may cause deletions or inversions. So deletions and inversions, like it can cause when this transposition events can take place, right? And or move the host sequence to a new location, or the movement of the host sequence to a new location also takes place. So both, like all of these three activities can be performed during a transposition events when it takes place. And the replicative transposition proceeds through co-integration, right? When the replicative transposition takes place, at that particular point of time, it proceeds through co-integration. So integration takes place at the same time when the replicative transposition is being taking place. So this, for this particular option, this particular question, option 2 is the correct one. B, C, D are the correct options here. Now coming to the question ID 228, right? So it says that the following statements are made with reference to the structure of the nucleosome. So here are the statements which are basically made in reference to the nucleosome structure. So first statement says that the histone tetramer in the core of the nucleosome comprises of H2A, H2B, H3 and H4. Then it says that the second option says that the N-terminal tails of the core histones are believed to stabilize the 30 nanometer fiber of the nucleosomal DNA by their interactions with the adjacent nucleosomes. Then it says that the post-translational modifications of the N-terminal tails as well as the globular domains of the core histones modulate the transcriptional events. Last option, it says that according to the zigzag model of the 30 nanometer fiber, the linker DNA circles around the central axis of the fiber as the DNA moves from one nucleosome to the next. So out of these options, which options are the correct one regarding the structure of the nucleosome? So anybody has attempted this particular question can answer me in the chat box what option they have opted for. Anybody? Okay, Bhavapriya, she says option 2. Anybody else who have attempted this particular question? Jerina, Gabriel, she says option 2. Okay. So most of you are saying option 2. Keetana, she says option 1. Right. So coming to the explanation. So Keetana, you are right. Option 1 is the correct one here. So the explanation, it says that the histones H2A, H2B, H3 and H4 are known as the core histones, right? So these are the core histones present, and histones H1 and H5 are known as the linker histones. These two are the linker histones. The core histones all exist in dimers. So histones, the core histones, all the core histones, they exist in a dimeric form. And the four dimers come together to form one octameric nucleosome core, right? So the linker DNA circles around the central axis as the DNA moves from one nucleosome to the next in the solenoid model. So in case of solenoid model, what happens? This linker DNA, the linker DNA, that is the H1, right? The linker DNA, it circles basically around the central axis as the DNA, it moves from one nucleosome to the next. So for this particular question, the option 1 is the correct one here. First, that the histone's tetramer is the core for the nucleosome comprises. It basically comprises of H2A, H2B, H3, and H4. And all of these core proteins, these core histone proteins, they are present in dimeric form, right? And B, it says that the N-terminal tails of the core histones are believed to stabilize the 30 nanometer fiber of the nucleosomal DNA by their interactions with the adjacent nucleosomes. This particular option is also correct one here. And then it says that the C option, it says that the post-translational modification of the N-terminal tails as well as the globular domains of the core histones modulate the transcription events, right? So these two options are the correct one here. Then, so option 1, whosoever marked have done it right. Now coming to the option question ID 231. So here it says that the precise recognition of the tRNAs by their cognate aminoacyl tRNA synthetases is crucial for the fidelity of the protein synthesis, right? So in the context of the aminoacylation of the tRNA alanine, with its cognate aminoacyl tRNA synthesis alanine RS, and based on the studies on the molecules of the E. coli origin, following statements are being made. So first statement, it says that the anticodon of the tRNA alanine makes important contribution to the specificity of its amino acylation by alanine RS. Second, it says that the mutational analysis have shown that for amino acylation of the tRNA alanine by alanine RS, the presence of a wobble pair in the acceptor stem G3 is to U70 is most crucial element. C option, it says that the amino acylation of the tRNA alanine by alanine RS occurs even if the anticodon of the tRNA alanine is mutated, right? Then last option, it says that a microhelix lacking a clover leaf structure and harboring only the acceptor stem sequence of the tRNA alanine is specifically amino acylated by alanine RS. So which of the option would be correct here? Yes, Jelena and Gabriel, since they haven't mentioned that it is present in dimeric form, therefore it is incorrect one. So Kirtana says option C. So you have to basically pick which option is the incorrect one. Right? So anybody who have attempted this particular question? Okay, you have changed your answer. And Kirtana, option D, now you say. And anybody who have attempted this particular question? Then we'll be coming to the explanation and we'll be knowing whosoever mentioned the correct answer. So coming to the explanation, the option A is the correct one here, right? But the incorrect option is the option A. That is, the anticodon of the tRNA alanine makes important contribution to the specificity of its amino acylation by alanine RS. So the explanation here, it says that across a range of organisms from bacteria to humans, the primary determinant of tRNA recognition by the alanine tRNA synthesis is a single GU base pair, right? in the amino acid arm of the tRNA alanine. A short synthetic RNA with as few as 7 base pairs arranged in the simple hairpin mini helix is efficiently amino acylated by alanine tRNA synthetases as long as this RNA contains the critical GU sequence, right? So anticodon of tRNA alanine makes no contribution to the specificity of its amino acylation by alanine RS. So this anticodon tRNA alanine, it is not responsible for the specification. So there is no contribution. It has no contribution in the specificity of its amino acylation. So this particular option, option A, is the incorrect one here. So whosoever have marked option A have marked it right. Okay, don't be biased. You haven't attempted this particular question because it was lengthy. Okay. So this much are you clear with why option A is the incorrect one here? So coming to the question ID 232. So here is a figure. In this figure, it is being given that the replication of the DNA beginning from the origin of the replication of the chromosome of a newly identified bacterium having a double-stranded circular DNA genome is being shown here, right? So the characterization of the DNA polymerase responsible for the genome replication showed that the DNA synthesis occurs in 5' to 3' direction, and it depends on the presence of a primer, right? So primer is responsible here, as in case of Escherichia coli. Now polarities of the DNA 5' or 3' are being shown here as such. So polarities is being shown here. This is the 5' to 3' leading strand. This is the lagging strand here. It is being given. Now it says the replication begins at a point marked O. So here is the origin of replication where the replication basically begins, right? On the left of the bubble, and both the parent strands were replicated concurrently. Now the longer arrow inside the bubble, it shows the leading strand. So this is the leading strand here, it is being shown, right? And whereas the shorter arrows marked A, B, C show the Okazaki fragments. Now in this particular model, the A, you have to identify what A basically depicts here. So first option, it says it depicts the bidirectional mode of replication wherein synthesis of the Okazaki fragment marked C occurs prior to those marked A and B. Second, it says the bidirectional mode of replication wherein synthesis of Okazaki fragment marked A occurs prior to those marked B and C. Third, it says the unidirectional mode of replication wherein synthesis of Okazaki fragment marked as C occurs prior to those of marked A and B. And last, it says that the unidirectional mode of replication wherein synthesis of the Okazaki fragment marked A occurs prior to those marked B and C. So which options is the correct one here, which represents A? Anybody who have attempted this particular question? So Bhavapriya, she says option D, unidirectional mode of replication, wherein synthesis of the Okazaki fragment marked A occurs prior to those marked B and C. So basically in Okazaki fragment also, the synthesis basically takes place from 5' to 3', as everybody knows. So let us see which option would be the correct one here, right? Firstly, which one will take place? C will occur or A will occur? So here it is being given that option C is the correct one here. So option C, it says that the unidirectional mode of replication wherein synthesis of the Okazaki fragment marked C, it occurs prior to those marked A and B, right? So this particular Okazaki fragment, the synthesis of this fragment will take place before B and A, right? So here it says that the DNA explanation, it says that the DNA synthesis takes place simultaneously but in opposite direction on the two DNA template strands starting from the origin, right? So double-stranded DNA molecule unwinds to provide two single-stranded templates to form replication bubble. Now bidirectional replication is expanding of the replication bubble in both the direction, wherein the unidirectional in one direction. In the given figure, it is being expanding only in one direction from the origin. So this represents unidirectional replication. So it is a unidirectional kind of replication, not the bidirectional one. Also, one strand is synthesized continuously and the other is discontinuously. As we know, the continuous one is the leading strand and the discontinuous one is the lagging strand. So the continuous strand or the leading strand is the one which in which the 5' to 3' synthesis proceeds in the same direction as the replication fork movement, while the discontinuous strand or the lagging strand is the one in which the 5' to 3' synthesis proceeds in the direction opposite to the direction of the fork movement. Now, the DNA synthesis on the lagging strand continues until the fragment extends as far as the primer of the previously added Okazaki fragment is there. So a new primer is being synthesized near the replication fork to begin the process again. Hence, the order of the Okazaki fragment in this particular figure, it would be C, B, and A, right? So firstly, this particular here the primer would be there, and from here the 5' to 3' direction the Okazaki fragment would be formed. Then the B segment would be formed, and after that A would occur. So order would be first C, then A, then B, then B, then A, right? So option C would be the correct one here. So coming to the question ID 233, which says that which one of the following statements is not a correct feature of E. coli RNA polymerase, right? So statements regarding RNA polymerase is being given and you have to pick which of the following options is not the correct one. So this is the prokaryotic RNA polymerase here, it is being mentioned. So first statement, it says that the presence of the sigma subunit along with the alpha 2, beta, beta dash, and omega core RNA polymerase is required for its promoter specific binding. So sigma subunit is involved and this particular core protein is involved. So all of these proteins together, they make the holoenzyme, right? And it is being required in its promoter specific binding. Second option says that the presence of the sigma subunit along with this core factor, this core protein of RNA polymerase is not necessary for the core RNA polymerase to bind to the DNA template. So coming to option C, it says the mutations in the B subunit in its rifampicin resistance defining region RRDR. It confers the rifampicin resistance phenotype. And option D, it says that the mutations in the sigma subunit in its rifampicin resistance defining region RRDR, it confers the rifampicin resistance phenotype. So which one of the statements are the incorrect one here? You have to answer. So, okay, Bhava says she hasn't attempted. So Jerina Gabriel, she says option C. Okay, option C is the incorrect one according to you. So yes, anybody else who has attempted this particular question? So yes, so option D is the correct one here. So whosoever have marked option D, this is the incorrect statement. Option D is the incorrect statement here. Rest all the options are correct. So how option D is the incorrect one here? So it says that the RNA polymerase, RNAP, in case of Bacterium Escherichia coli, it exists as an approximately 400 kilodalton core complex consisting of two identical alpha subunits, similar but not identical, beta and beta dash subunits, right? And an omega subunit. The core RNA polymerase associates with a specific protein factor. So all these alpha, beta, beta dash, right, and omega, these form the core protein. So two alphas are present. So two alpha, beta, beta dash, and omega, these form the core protein. Along with it, there is another special protein that is involved like with the core protein. It associates onto the DNA to start the transcription to take place. So the core RNA polymerase associates with a specific protein factor, which is the sigma factor, that helps the core enzyme recognize and binds to the specific deoxynucleotide sequence of the promoter region to form the pre-initiation complex, right? So mutations within the RPOB gene encoding the beta subunit of the RNA polymerase. It conferred the different levels of rifampicin resistance. So if there is a mutation which takes place in the RPOB gene encoding for the beta, this gene basically encodes for the beta subunit. If there is a mutation which takes place in this particular gene, then what happens? The RNA polymerase, it confers different levels of rifampicin resistance, right? Different levels of rifampicin resistance would occur due to this mutation. Rifampicin RIF is a member of rifampicin antibiotic used in the treatment of the tuberculosis and other bacterial infections. It targets bacterial RNA polymerase by binding near to the polymerase active site and inhibiting the elongation of the nascent mRNA, right? So to prevent any kind of like bacterial infection in our body, if we have this rifampicin antibiotic, this is a kind of antibiotic, right? So what does it do? It basically targets the bacterial RNA polymerases by binding near the polymerase active site and inhibiting the elongation of the nascent mRNA. So it basically inhibits the process of elongation. The catalytic core of the bacterial RNA polymerase consists of the alpha 2 beta beta dash omega chains, and the rifampicin, it binds to the pocket formed by the beta chain, right? And numerous mutations in the RNA polymerase beta chain, it confirms that the rifampicin resistance RIF or in Escherichia coli and Mycobacterium tuberculosis. So these mutations are clustered in 4 regions of the RNA polymerase beta termed as RIF resistance determining regions. So there are different regions. According to this regions, basically the mutation, the mutations within it basically defines that which level of mutation is basically taking place. So based on these findings, it is being observed that the option 1, option 2 and option 3 are correct, wherein option 4 is the incorrect one, right? So mutation sigma subunit in its rifampicin resistance binding region RRDR confirmed the rifampicin resistant phenotype. So this is the wrong one. Only in the beta subunit, basically, when this rifampicin antibiotic or the resistance takes place, then it confirms the rifampicin resistance phenotype. And option A is the correct one because we know that the core protein is alpha 2 beta beta dash omega, and along with that, when the sigma subunit binds to the DNA template and the initiation of the transcription basically occurs. And option B, it says the presence of the sigma subunit along with this core protein of RNA polymerase is not necessary for the core RNA polymerase to bind to the DNA template. So option D is the incorrect one here, right? So whosoever have marked option D, they have done it right. Now coming to the question ID 289. So it says for the template sequence given below, which one of the following combinations of the primers can hypothetically be used to amplify the target region? So you have to ignore the TM and the length of the parameters for the primers, and you have to answer for the template sequence given below, which one of the following combinations of the primer can hypothetically be used to amplify the target region. So this is the template that is being given to you, and this is the primer 1 and primer 2, and you have to tell me which option is the correct one here, which primer that you can utilize to amplify the target region. Has anybody attempted this particular question? Yes, I'm waiting for the answers. Which primer 1 and primer 2, which option would be the correct one? And it would be used to hypothetically amplify the target region. And the template sequence is being given here. Okay, Keetana says option B. Anybody else who have attempted this particular question? Let us see what is the explanation for this particular question. What is the right answer? So option B is the correct one. So Kirtana, you're right. So here is the explanation for this particular question. It says that the DNA is double-stranded, having two complementary strands, as we know, 3' to 5' and 5' to 3'. So primer 1 attaches to the 5' to 3' DNA strand near its 3' end, right? So primer 1, it will basically attach the 5' to 3' end of the DNA. So primer 1, basically it will be attaching here, right? At the 3' end of the target DNA, and from 5' to 3' it will be moving. So in this complementarity, which of the following options you find would be the correct one? So yeah, so it says the primer 1 attaches to the 5' to 3' DNA strand near its the 3' end. So primer 2 attaches to the 3' and this particular primer 2, it will attach to 3' to 5' DNA strand at its 3' end, right? So here it says that the primer 2, it will be attaching to this particular end and it will be moving from 5' to 3' direction. So accordingly, we can see which option is the correct one here. So B option is the correct one here. It says. So here it says that the 3' end of the both primers are pointed towards each other, and the terminated intermediate DNA region could be amplified. In question, only a 5' to 3' single strand of the DNA is being given. Right, only a 5' to 3' single stranded DNA is being given. The other strand should be 3' to 5'. So if we make complementary of this particular strand, the complementary region, which would be from 3' to 5', right? So here would be TA, GC, TA. So complementary if we make here, right? So here it would be T, here it would be A, here it would be G, C, T, G. So in this manner, if we make the complementary strand, so what happens is, here it will be the 3' end, and here would be the 5' end. From 5' end, if we see, it would be C, T, G, right? Here it would be G, here it would be C, here it would be A, here it would be T. So if we see that the primer 2, it basically binds to which region? It will bind to 3' end, right? So it will binding to the 3' end from 5' to 3' region. So from here it will be binding to, it would be here A, T, C, G, A, C. So A, T, C, G, A, C sequence would be here. So this particular primer 2 would be binding here, and primer 1, it would be binding here, right? So for primer 1, from 5' to 3', it would be C. Here it would be complementary to G. So it would be C, T, G, C, A, T. So in this manner, primer 1 would be binding here. In this manner, here from here to here, primer 2 would be binding here. So option B would be the correct one here, right? So in this way, you have to answer this particular question. The other strand, it should be 3' to 5' and complementary to the given strand. So therefore, the primer 2 will be binding to the 3' to 5' DNA strand and should be identical to the sequence given at the 5' end. As primer 2 sequence 5' to ATCGAC 3', given in the option 2, resembles the given 5' to 3' sequence. Therefore, option 2 is the correct one here. So did you get how this particular question would be solved? Firstly, you have to make a complementary strand here. Accordingly, you have to fit in which complementary primer would be attached to which region. So primer 2 would be attached to this particular site from 5' to 3' region, and primer 1 would be attached from this in this direction from 5' to 3' region. So option 2 would be the correct one, which will be showing the correct complementary stretch for this particular DNA, template DNA, right? So option B would be the correct one. So whosoever have marked option B, they have done it right. So we have come to the end of this question answer discussion for the CSIR NET paper 2021, which was held. I hope everybody would have understood and have understood what is the explanation for each and every question. So for both shift 1 and shift 2, I have explained it. So next we'll be coming with next unit, that is for unit 4. So there we'll be having a discussion about what are the explanation for the questions that came from unit 4, right? So this much are you clear with? You can go through this again, and from the standard book itself you can find out the explanation is being given here. And I have also mentioned, like from like page number also I have mentioned one of the question, and the tables also I have mentioned. So you can go through the books again. Standard books are the must. So go through those questions again. Wherever you have done the mistake, try to observe them and definitely try to improve for the next time, because two more exams are being coming now for the CSIR, and I want that all of you can like improve at that particular exam and clear the CSIR net exam in this particular year itself, right? Okay, thank you very much.